start speaking in Spanish. Ok, buenas tardes a todos los que están conectados para este seminario de Frontiers in Genomics. Es el penúltimo del año. Y como saben, las preguntas se plantean al final. Y bueno, ya sea que alcen la mano aquí y les dé la palabra, o poner las preguntas en el chat y ya las leeré yo porque ella no puede ver el chat. Pero bueno, entonces voy a presentar a la profesora invitada del día de hoy. Para mí es un gusto presentar a la doctora Julia Salzman, que es profesora asociada del Departamento de Biomedical Data Science, del Departamento de Bioquímica y del Departamento de Estadística de la Universidad de Stanford, en California. La doctora Salzman hizo su licenciatura en Matemáticas en Princeton en 2002 y su doctorado en el Departamento de Matemáticas en Estadística en la Universidad de Stanford hasta el 2007. Desde 2007 al 2008 fue Assistant Professor en Columbia University. Desde 2008 al 2009 hizo un postdoctorado en el grupo de Patrick Brown, que es muy conocido por los biólogos moleculares, en el Departamento de Bioquímica de la Universidad de Stanford. Y del 2009 al 2013 fue Acting Associate Professor, también en el Departamento de Bioquímica en Stanford y en el laboratorio de Patrick Brown. A partir del 2013 al 2020 fue Assistant Professor en el Departamento de Bioquímica de Stanford, y 2020 a la fecha, como ya les había dicho, es Associate Professor en el Departamento de Biomedical Data Science del Departamento de Bioquímica y el Departamento de Estadística de la Universidad de Stanford. La doctora Salzman ha recibido distinciones y reconocimientos importantes y solamente voy a mencionar algunos. Bueno, hice un resumen. En el 2002 fue Princeton Magna Cum Laude y High Honors en Mathematics. En el 2012 recibió el Pathway to Independence Award. En el 2014 recibió el Alfred Sloan Foundation Research Fellow in Computational and Evolutionary Molecular Biology. En el 2014 el Donald y Amelia B. Baxter Foundation Faculty Scholar Grantee. En 2015 el McCormick-Gadian Fellowship. En 2016 el NSF Career Award. Y hoy nos va a hablar sobre nuevos algoritmos que le permiten descubrir funciones biológicas en general en diferentes organismos. Y con eso, Julia, thank you very much. Now we are ready to hear your talk.
Well, thank you so much for the Yes, thank you so much. And I'm really excited to be here today to tell you about some recent work that I think is really wide-ranging and very exciting. So you may know the field of archaeology, and you may know it by the way that archaeologists approach classical study through what I will call customized digging. Recently, the field of archaeology experienced a technological breakthrough, which is the use of ground-penetrating radar. And this is a technology that allows archaeologists to rapidly survey large areas without the need to pre-specify or choose particular types of digging equipment, digging depth, and it works for any kind of archaeological site without specifying the particular pattern of an ancient civilization that we were looking for. Okay, so this idea, I think, inspires a question for genomics, which is, can genomics develop something like ground-penetrating radar? And it's, of course, an aspirational vision, but what would we like from that aspiration? It would be a technology that would enable us to analyze large amounts of data very rapidly without pre-specifying a specific bioinformatic custom tool. And it should work for discovering any kind of novelty, a new mechanism, for example, a biochemical mechanism. It could provide an approach for pathogen surveillance, or to discover potentially master regulators in cell biology or genetics. So what I'm going to tell you about today is why, first, to give you a few minutes of why we might aspire to this kind of ground-penetrating radar for genomics. That is, what are the missing and limited inferences that are available with current approaches. I'm going to tell you about what NOMAD is, which I think it is an interesting step towards a ground-penetrating radar for genomics. I'll say that today, NOMAD is not one thing, which is, I think, a very well-studied area, which is quantifying gene expression. Although we have ideas for how to extend NOMAD in that direction. Okay, and then finally, I'm going to focus on NOMAD's prediction of new biology, which to me is some of the most exciting. NOMAD has made some really exciting predictions for single-cell RNA sequencing. From single-cell RNA sequencing that has revealed new biology that has been missed. I'm going to tell you about how we've used it to identify variants in SARS-CoV-2, and also some very exciting unpublished work in tuberculosis. And I should say all of it except B is unpublished. Okay, so what I want to first say is that I want to define what I mean by a reference and a reference genome. And a reference is simply a string of characters, ACGT, that is supposed to represent the base pair composition of the human genome or another organism's genome. And how are references used in bioinformatics today? Well, they are used as the primary object of inference in this following sense. Bioinformatic algorithms, with very few exceptions, take raw reads that are observed from a sequencer, and the first step is to align them to a reference genome. So we have the reference sequence, we have a read. And the first step is going to be to align this read to a position in the genome, which reduces the raw data to it's a transformation of raw sequences to positions, and possibly mismatches are included in this reduction, possibly splice alignments, if you're familiar with that. But because this is a procedure of transformation, it introduces errors. In addition, sequences that are absent from a reference are ignored. And this has enormous implications for health disparities throughout the world, because the vast majority of reference genomes are built on people from European ancestries due to the amount of resources that have been poured into endeavors to sequence human genomes being directed at people of European descent. And here's a great example of this. In a set of only 910 individuals of African descent, about 300 megabases, that is about 10% of the human genome, was found to be missing. And that means that if people are studying African Americans or Africans, and they use the human reference genome as their in a bioinformatics study, they will miss any variation in any of the genes because these sequences are simply not present. Another major limitation of reference genomes comes from an assumption that is simply not true. And that assumption is that each cell in your body has the same genome. A really extreme example of how this fails to be true is in the context of cancer. Tumors have long been known to be aneuploid, and recently they were discovered to undergo a process called chromothripsis, where chromosomes literally are shattered, and they are then stitched back together in a totally different order than appears in the reference genome. And any kind of analysis of tumor RNA-seq, for example, would fail to find sequences that are derived from this stitched back together piece, for example, BI, because BI is not part of the original reference genome. And I would say that while we have examples like this, positive examples of cases like cancer where we know the reference genome deviates from normal, this phenomenon could be much more prevalent. Okay, a final example of why we want to move away from reference genomes comes from the viral and bacterial world. The microbial world overall has incredibly plastic genome biology, and that has been revealed by multiple groups. So phages, transposons, mobile DNA, including horizontal gene transfer, and perhaps other mechanisms that are yet to be discovered, modify the genomes of even extremely common human pathogens or common commensal bacteria like E. coli by inserting themselves into the genome. And when they do this, for example, in this case, a phage inserts into the E. coli genome that has a gene called the shigotoxin gene. That shigotoxin gene is made by E. coli and causes diuretic disease. Okay, so mobile genetic elements are extremely important in any kind of phenotypic study, and they are so extensive that it would be impossible or infeasible to store and search all possible genetic variants in the microbial world. Moreover, it's indirect, as I described to you. Okay, the second major limitation of genomics today is its custom nature. And what I mean by this is that each of these examples, microbiology, your fields, microbiology, immunology, human genomics, they use their own specialized bioinformatics software directed at identifying very specific features in a genomic context. For example, in microbiology, detecting mobile genetic elements. In immunology, detecting VDJ recombination. In human genetics, detecting alternative splicing, a very specialized algorithm. And this algorithm will not at the same time detect RNA editing, which is another important way that the human genome is regulated. So altogether, this results in a specialized zoo of algorithms that are used in genomics today that simply don't generalize, they're too time-consuming, and they lack a statistical foundation to make their calls analyzable in the context of what we care about, which are things like false discovery rates or p-values. And as I hope I'll tell you about later and convince you of, these biases are not just technical, sort of technical notes. They actually limit the biology that we can discover with genomics today. So we know kind of by theoretical analysis of what alignments and bioinformatic algorithms do, that they will miss some of the most important and interesting genomic novelty that exists, such as expansions of repeats, which we know cause some neurodegenerative diseases, structural variation, insertions of mobile elements, and anything that any kind of mechanism or variation that we don't know about yet, which are arguably the most important and interesting tasks for genomic approaches. So is there a way forward here? If we want to develop ground penetrating radar for genomics, we need a model that is something like what archaeology has. So we need some kind of pattern underground to search for it. For us, it's not underground, it's in genomic sequencing data. So let's think about, is there a pattern that we might be able to search for and recognize? And take 3 case studies. One is one of the most fascinating kind of genomic regulation that occurs in human cells, which is how the adaptive immune system evolves. So it evolves through a process called VDJ recombination and somatic hypermutation, where a fixed locus in our genomes gets rearranged and recombined and mutated to generate antibodies. So in VDJ, one of 39 variable regions is paired with one of 27 diversity generating regions. And then it is yet a third set of the joining region is chosen. These are then carried forward in every mature B cell or T cell, and they are the template for antibody production. And they have a feature, which is very interesting. They are hyperdiverse, these loci are hyperdiverse between cells, but within a single cell, they are nearly clonal. Let's take a second case study, and that is in a totally different context, the microbial world. Each of these lines represents a strain's genome. This triangle is just here to reflect a constant region of DNA that isn't changing, possibly the ribosome, a ribosomal protein. these circles reflect mobile elements, and they jump around between genomes, insert themselves, and remove themselves. Now, this case study also has a similar similarity, which is that it is hyperdiverse between strains, but within a strain is nearly clonal. And then final case study, something, one of the most interesting parts of the way that the human genome is decoded and enables multicellular organization from a single common DNA sequence is alternative splicing that affects more than 95% of all human genes, where one exon, the blue, could be spliced to a purple or an orange. Splicing is developmentally essential. Its dysregulation is known to cause diseases from cancer to neurodegenerative diseases, and it has a similar feature that it is very diverse between cells, and within a cell is nearly clonal, maybe not exactly clonal. So I'd like to ask whether we can use these case studies to define something like a pattern that would enable us to develop ground-penetrating radar for genomics. And what I'll tell you about is a step in this direction, which is NOMAD. NOMAD is a simple statistical and reference-free approach developed by a team of really talented individuals, Caitlin Chang and Tavora Baharov, developed the first version of NOMAD, and then Ruzbe Denganaziri and George Henderson, along with others, have explored it and developed it further to make it really interpretable and generate some of the most exciting discoveries, I think, that it has made so far. So I'm going to return to the case studies and try to be a little bit more quantitative and concrete here. So what if we didn't have a reference genome, and perhaps we didn't even know that alternative splicing existed? How could we identify or formulate a feature of sequences, and sequences alone, raw reads that came from alternatively spliced transcripts? Well, as this color diagram shows, alternative splice variants are unified in their feature of having a constant sequence, blue, followed by a variable sequence. If exon 3 is used, that variable sequence is pink, and if exon 2 is used, it's yellow. Okay, and so at the level of nucleotides, if we're able to stream in reads, we would see that when we saw this former GAAT, sometimes it would be followed by one sequence and sometimes by another. And I'm using these formers as toy examples, and of course, we would need a longer sequence context, but I hope you bear with me. Okay, similarly, what if we were trying to develop a method to identify mobile elements? Well, the end of the mobile element has a feature that it is always constant when you observe it, but if the element is actually mobile, then sometimes its neighboring sequence will be in the middle of the genome, ATCG, and sometimes it will be near the end, resulting in a different sequence. Similarly, antibody diversity is generated by DNA splicing that will have the same feature. The constant region of the antibody chain, GGAA, will be flanked by a set of different variable regions. And maybe a final example here is something that would be less dramatic, but seen in viral strains or microbial strain variation, where a single nucleotide could define a strain. And in that case, there would be a constant region, box in blue, followed by two variable regions that would differ by a single nucleotide, but yet they would be very reproducibly different. And I don't have time to tell you about and enumerate how many different important mechanistic molecular processes have the same feature of being defined by a set of sequences where there is a single sequence that is constant followed by a set of other variable sequences. Okay, so we need just one more piece to define what this underground feature is for a ground penetrating radar. And that is, it's not enough just to have a sequence in blue followed by a set of variable sequences, pink or yellow. And this is a really critical point. In order to identify biologically important variation, we are interested in cases where the sequence that is pink or yellow, which we call the target, is a function of the sample. Okay, so from now on, I'm going to call the blue sequence an anchor and the variable, which is constant, and the variable sequence a target. So formally, what we're going to be looking for are cases where the probability of observing variant 1 is a function of the sample. So essentially, if I told you I'm observing reads from this macrophage, for example, once I tell you I'm observing reads from a macrophage, I can very well predict the variant that is going to be downstream of the blue sequence. Whereas if I tell you I'm sampling from a dendritic cell, I can also tell you very well with high probability what the sequence is downstream of the blue. It's a different sequence. Okay, so this is essentially, it's an intuitive idea, but it is the heart of Nomad, and it enables a reference-free algorithm for discovery. So that's essentially the conceptual background and definition that allowed us to develop an ultra-efficient, reference-free approach for genomic discovery, taking raw reads, performing a pre-processing step, which allows us to look for every single k-mer in the sample, define an anchor for it, catalog all of the variation in its targets, perform a statistical inference to tell us which anchors have this sample-specific sequence diversification, and then perform a statistical assembly from these anchors and interpret them. We can sometimes interpret them using a reference genome alignment, a partial genome alignment, or bypass alignment completely and perform, for example, structure prediction or protein prediction from in silico translation. Okay, so Nomad is a very, very simple concept, but to make it work, there is a lot of hard work that has gone into this, from new statistics to new software engineering. Okay, so I want to tell you a little bit about the statistics behind Nomad, because it is really the reason that we are able to perform the inference that we can. In the simplest null hypothesis, we formulate targets are drawn from the same distribution across samples. And these samples could be very different biological entities. Here, I'm going to explain it to you for a case of samples being 4 people, each of them infected with perhaps a different variant of a virus. The variants are depicted, are illustrated by being variant 1 is orange and variant 2 is pink. So we want to test whether the variation in the number of these different targets per person can be explained by chance. That is, explained by sampling from a null distribution. Okay, and so in order to do this, we needed to develop a new statistic because the ones that are available today are extremely common. They have important properties and are of course very useful, but they were developed by very famous luminaries in the field of statistics, but they were also developed for a different era of data. They were developed in the 1900s for classical data that was not as noisy as the data that we observe in genomics today. And here's a really important motivation for why we need new statistics. I have generated, or actually Tavor generated, this heat map, which is essentially, we would all agree as biologists, not very interesting to study further. If I said, here's a list of targets are hidden on as the row names and columns represent samples, and I told you, aha, I found some interesting biological sequence here, you would probably say, I don't want to spend my time investigating this. Whereas in this example, we would probably agree there is a signal, and especially so if I told you this group of people, this group of samples were people that had a different, let's say, constellation of clinical symptoms than these people. Chi-squared cannot distinguish these scenarios. So we decided to develop a new statistic that had important properties. We wanted one that was computationally efficient and gave us precise p-values, that was robust to biological noise, being defined as examples like these, and had the power to detect biologically important signals. Okay, so let's walk through what is NOMAD statistic. Okay, it takes a set of targets for a fixed anchor, the observations per sample, and asks the question, do the target distributions come from the same distribution, or are they sample specific? And in order to test this, we compute the total number of counts per sample. We generate a function on the targets. We can do this but randomly or in a more disciplined way. We then compute, so this induces a function on the target space and gives us a way of summing up counts. We sum up the counts in the table that are assigned the value 1 and take their weight, the average compared to all counts. We then compare for each sample the difference between the estimated value of mu and the global mu, if we basically the global mu being the total number of counts across all of the tables collapsing across sample identity, weight that by the number of observations per cell, and then either randomly or using metadata, define another function on the columns to combine the SJs to get our test statistic. And what you can just take away, that was the last amount of math for those that not that interested in it. What's great about NOMAD statistics are that they have closed form p-values and are robust. So what we did is a simulation where we took random samples from the same distribution. These are random samples. Each column is a random sample from the same distribution. And then added noise, which you could think of as just kind of adding sporadic counts to each cell and dial up that amount of noise from 0 to 1. So chi-squared starts saying this is a signal as soon as we add a little bit of noise, whereas NOMAD is much more robust to this. And that has a really important feature that allows us to prioritize biological signals in the presence of noise. Okay, so for the rest of the time, I'm going to explain the way that we have applied NOMAD to a variety of biological contexts for discovery. And I want to start with NOMAD's application to single-cell transcriptomics. So NOMAD can be applied, I just want to emphasize, it is an algorithm that has no pre-specification about what mechanism, what data type. It could be RNA-seq, it could be single-cell RNA-seq, could be DNA-seq, and probably even protein. So in everything I'll tell you about, we are using the identical algorithm, no tuning parameters. We decided to investigate the variation in the human transcriptome in primary tissues through using the Tabula sapiens atlas, which is a multi-organ single-cell atlas. And what I'll tell you about is a study of more than 12,000 cells in 18 tissues. And one thing that is quite interesting to me about this is NOMAD was run with a subsample of all of the data that was in this atlas. There was no cell metadata used. That is, we didn't know anything about cell type. There was no annotation done. And we did not know anything about the genome before statistical inference was made. Okay, so we take NOMAD outputs, we find these significant tables which correspond to an anchor sequence. We then grow this anchor sequence in potentially multiple ways using something like a de novo assembly, but one that is statistical, and I think that's part of the key to the findings. We then analyze these statistical microassemblies and classify them into different buckets. For example, we might be able to say that this anchor was called because it represents splicing variation between cells, or because it represents different non-coding RNA variation. We're going to start with splicing because splicing is very well studied. Many people have published, true positive controls for benchmarking the analysis. There have been deep biological studies by people showing that some isoforms are very important, and there are very well-developed computational tools for annotating isoforms in the human transcriptome. So we started by asking whether when Nomad calls an anchor sequence and it represents what we think is splicing, is that splice junction previously annotated? And if we had run star, which is a popular aligner, on the raw reads, would star have also called the junction? And we see that when we look at the most highly abundant sequences, as we would hope, the vast majority of them are both annotated and called by star. But a sizable majority are annotated, but were actually missed in raw star reads, which means that any spliced alignment program, depending on star, would have missed these as well. Oops. Okay. And I want to be clear that the annotation here is very comprehensive. It's based on computational prediction. And here's an example of one gene, Gas5, and the number of different isoforms it has. As we relax the threshold on the number of reads that we're observing for each junction, we start to see interesting features emerge. For example, ones that are annotated but not detected by star. I think I actually misspoke and I apologize. This blue bar earlier, star detected both bars. It's just that in the blue case, the isoform was not annotated. Whereas once we relax the absolute numbers of reads, we start to see examples of where star did not report the junction, but it is annotated, which is strong evidence that we are detecting a true positive and that star had a false positive. A false negative. As we relax the threshold more and more, we see different abundances of categories, including cases where star failed to detect the annotation and was not annotated in the reference. Okay. We also assessed Nomad's specificity by using the fact that in this atlas, there are replicates of the same tissue observed in different donors. And I want to be clear here that when I say there was the same tissue, it was actually a subsample of a tiny number of cells, like 400 cells for each tissue. They were not matched. So the composition of the different cell types within each donor are different. And we would not necessarily expect that when run on donor 1 or donor 2, that we would have the same genes called. And in the red, you can see the calls by Nomad in donor 1 and donor 2, as well as the number that are shared. And this number is highly enriched compared to the other algorithm that we're comparing to, Splice, which was actually one that our group developed just a few years ago. Okay, the same kind of enrichment for reproducibility is found in Nomad in other tissues, including in the blood and in the muscle. So we decided to take a look at one of the predictions that was like basically a Nomad call that was missed by Splice, but reproducible in all of the muscle cells. And it was Gas5, which is a very interesting non-coding RNA. And Nomad calls 3 different variants of this gene. They're depicted in different colors here. And even though it had no cell type metadata to begin with, when we investigated the fractions of these splice junctions in different cell types, we can see that there is remarkable concordance in reproducibility. And that the effects of, in terms of like the fractional difference of the different isoforms in different cell types is large for alternative splicing. Here, the green isoform comprises about 50% of the transcripts in endothelial cells and about 20% in of the red, which is a large shift in the field of splicing and really suggests something very interesting could be going on. The shift is even more extreme in T cells. Okay, here's another example. CD47 is a very well-studied and incredibly important gene for human immunity. It is the target of multiple drugs that have implications for cardiovascular health, and it's a critical cancer immunology target. Okay, it has several different annotated splice variants, but overall, people don't understand which splice variants are expressed in which cell types and how they're regulated. And moreover, there has never been a report of these two minor splice variants, the orange and dark, like light and dark orange, at least to my knowledge, that Nomad predicted. And so you can see, based on this coloring scheme, this is a cytoplasmic tail of the protein. This is its IG domain. Each of the splice variants is predicted to modify one of these important signaling domains or extracellular domains, including the novel isoforms. So this is a new discovery by Nomad, and you can see that one of the interesting features is that, again, without knowing anything about the cell type metadata, Nomad has found cases of where a particular compartment, in this case stromal cells, have almost exclusively express variation in the, they express the light blue or dominant isoform. And there are some rare cells, 4 of them, that express only these novel isoforms. Now, this I say is rare, but they're 4 out of about 400. So that's actually not so rare if you think about the human body. That's 1% of cells, which could have an enormous impact on understanding the biology of CD47. You know, I'm not claiming it necessarily does, but it could. Here's another example of a discovery that Nomad made in the gene RPS24. RPS24 is an essential ribosomal protein gene. It is conserved all the way to bacteria. And it has a very interesting microexon that is only 3 bases long. Nomad predicts the non-trivial abundance of this isoform, which has never been reported or not part of the annotation at least. And again, without knowing metadata, it also finds a pattern of RPS24 expression that is compartment specific, the blue epithelial cells having a very strong preference for this darkest, second most abundant isoform, whereas pink stromal cells almost always prefer expressing the green one. And just to hopefully entice you more about the importance of splicing, we did a computational follow-up here. This is work this is data from mouse, spatial transcriptomic data from mouse, brain, and kidney, where we're showing you the gene expression versus splice isoform expression. And if you focus on these 2 lobes of the brain, you can see that there is interesting gene expression change, but very little, very little splicing variation change, which shows that splicing is regulated in a way that is independent from gene expression. Okay. One of the interesting features of the Tabulosapiens Atlas is that its extensiveness allows us to investigate pan-tissue regulation. That is, we asked for each gene, how many times is it found across all of these tissues? We were interested in genes that were found in every single tissue or in nearly every tissue. HSP90A1 is a master regulator, a heat shock protein that was found in every single every single tissue. These genes are the ones that were found in N-1 tissues. And they have a remarkable feature and enrichment for being splicing factors or histone regulation. When we relaxed the criteria of the minimum number of tissues found to about 15 and did go-term enrichment, we also found something quite striking. Essentially, the only genes with high amounts of fold change and low p-values were genes involved in RNA metabolism, either processing or splicing. And again, if you're familiar with doing single-cell genomics, it is very rare to find this kind of gene enrichment in the most called genes. Usually, there's a bias towards highly expressed genes that are hard to understand. Okay, and then I want to give a final example of we investigated cases where NOMAD predicted very complex alternative splicing in a gene. And that was, as I said, it was enriched for splicing factors such as PRPF38B. We investigated the splice variants found, and you can see many of them are diverse and splice into an ultra-conserved or extremely highly conserved intronic region, including unannotated intron retention that we are planning to investigate further. Okay, so I want to give you one quick other tidbit of analysis that we did in humans, that is of genes, a locus that is highly repetitive in the sense that it is, there are more than 13,000 variants, most of them thought to be pseudogenes, of the U6 snRNA that is critical, essential for splicing. It is a non-coding RNA. And because of the number of repeats across the genome, STAR cannot map these, it cannot distinguish variants well, and many times actually fails to map at all. We used NOMAD to investigate whether specific variants of the gene were, or specific isoforms of this non-coding RNA were cell type specifically expressed or cell specifically expressed. And the columns here are cells, the rows are different targets, you could think of them, or microassemblies. And you can see that there is strong differentiation between cells and which microassembly is expressed in them. We analyzed the most abundant microassemblies and found in one case that they had very high similarity to RNU8 or unambiguous similarity. But what was interesting is they extended far beyond the annotated end of RNU68, implying that possibly this variant has a unique difference in the way that its 3' processing occurs. Another interesting example is in skin, where we have strong evidence that there is expression of a pseudogene, 6P, that includes the use of an upstream promoter. So overall, NOMAD, this is just an example of how NOMAD enables us to map and analyze non-coding RNAs. And we have similar and many other examples of this. Okay. And then finally, I told you that NOMAD is agnostic to the type of molecular mechanism it's detecting. And so indeed, without asking it to, NOMAD identifies cases of editing and hyperediting in many genes, including in the gene ANAPC16, which is an anaphase regulator. Overall, these are the microassemblies and their multi-way alignment that were detected in different donor tissue pairs. You can see that there's a signature of the canonical A to I editing and actually A to I hyperediting. For example, this sequence has 5 different edits and comprises a non-trivial fraction of the total transcripts from the locus. Overall, a minority of transcripts are actually non-edited. a similar example in AGO2, which is a famous gene that regulates microRNA targeting. Okay, so in summary, in this pilot study, which is a subsampling of cells and cell types, we have discovered novel isoforms and editing events, including a hypothesis that splicing factors in histone regulations themselves are involved in some autoregulation of their own splicing. The conservation of the intronic regions in these factors suggests a mechanism for regulation that is, of course, ongoing work. We've also discovered some interesting examples of non-coding variant expression that is cell regulated. Beyond what I've told you about today, NOMAD enables local MHC variant typing, finds examples of transposable elements that are inserted into the genome, like ALUs, and also somatic variation and VDJ recombination. So I think overall, I'm very excited about this because of both its biological discovery, its statistical transparency, and the fact that it is extremely scalable. So the speed that NOMAD runs is much, much faster than current algorithms that use star and downstream processing. Okay, so I want to spend about 5 to 10 minutes telling you about NOMAD's use for microbiology and emphasize again that the data I'm going to explain that we've analyzed with NOMAD was chosen randomly from the short read archive. It wasn't random in the sense of we decided what kind of biology we're interested in, but we didn't like pick data set 1 and analyze it with NOMAD and not find something, so moved on to 2. These are just saying, okay, this is an interesting data set, let's download it and analyze. So the first one is returning to the kind of intuitive, simple idea of what NOMAD has the potential to do, which is to analyze whether people are independent samples from the same set of variants for a specific anchor. In this case, we might be interested in cases where there's an anchor sequence followed by a mutation, because in the context of a virus like SARS-CoV-2, variants that are small, like single point mutations or insertions deletions, can have a large effect. So we ran NOMAD. We took a data set where it was known that patients had both Delta and Omicron variants, but we did not run the algorithm with any knowledge of which patients had which infections. There was no reference genome used, and we called significant anchors. Among the most significant, when we extended them with this microassembly, they revealed changes in the SARS-CoV-2 genome that were identical to ones that were annotated as being different between Delta and Omicron, including in regions that were called BA.4 and BA.5. And what's notable here is that the data was collected from patients in 2021. That was months before BA.4.5 and BA.5 were classified and cataloged by the WHO. And it implies that if NOMAD had been run and we had done this analysis, we could have predicted these variants. And I think that's very promising for the context of viral surveillance. We also performed a similar analysis of data scraped from the original genomic surveillance in South Africa that identified Omicron. And we performed another kind of analysis that totally bypassed knowledge of any reference genome. That was to take the microassemblies, assemble them, and predict what proteins that were enriched in the anchors that were called by NOMAD. And you can see what was very interesting to us is there's an incredible enrichment for NOMAD anchors called in the RBD, which is the receptor binding domain of SARS-CoV-2 in this context in France, and in the spike protein in South Africa. The other hits here that are enriched are provocative and partially known as being important in SARS-CoV-2 biology. Okay, so I'm going to turn now to very recent unpublished work in Mycobacterium tuberculosis done by Jordi Abante in collaboration with Alison Carey's lab at the University of Utah. TB is one of the most pressing human health threats worldwide. There's emerging drug resistance, and it is a huge burden on human populations. It is considered to be clonal and non-recombinogenic, which is part of the reason we decided to analyze it with NOMAD. There has been historically known to be a mobile genetic element called IS6100, and in the past has been used classically with DNA restriction fragment polymorphisms to define strains, which are currently classified into 7 lineages. These 7 lineages today are defined by SNPs. Okay, so we wanted to test the hypothesis whether indeed IS6100 was actually as ancient as we thought, and or whether we could use NOMAD to classify strains without knowledge of any patient metadata or strain typing. So we took 40 isolates that were randomly chosen from two sub lineages, so the closest known strains in TB. That this discrimination between 2.2.1 and 2.2.2 is currently done by assessing a set of 44 SNPs. Okay, so we asked, can NOMAD detect the transposons de novo? If there was movement of this element, we would expect it's the anchor sequence, which is the bounding element, bounding sequence of IS6100, to be neighboring different sequences in the genome. Okay, so there would be sample-dependent sequence diversification, which falls into the framework of what we expect NOMAD to be. So we ran NOMAD and investigated some of the anchors at the bounding elements. And what was very interesting is we were essentially able to predict sub-lineation extremely rapidly with no manual supervision at all. So we took the set of targets, and targets are the ones that are in red are annotated as being, or they can be aligned to these small sequences called targets can be aligned to the TB genome. The ones in green cannot be. Okay, so we formed this matrix that was called of elements called by NOMAD and performed naive unsupervised clustering. And interestingly, we were able to get more than 90% prediction. I would almost argue that the failures of the prediction are actually very provocative and interesting. And the clustering precision improved significantly when we included target sequences that were not part of the reference genome, okay, that all post facto, which is also very interesting. Then finally, we sought to test whether NOMAD could identify sub-lineation, that is, potentially identify strains of TB that may be emerging on the basis of the IS6100 movement that would be missed by traditional SNP-based approaches. Okay, and indeed, we found NOMAD calls this table as significant, suggesting that IS6100 is mobile within the lineage 2.2.2. And then when we investigate the clustering of the targets within this sub-lineage, we find some important clinical predictions, well, I should say important phenotypic predictions that are aligned with the targets. That is, purple sequences, ones that come from India, tend to have similar features of their target expression, suggesting that these strains are evolving after a bottleneck that involves mobility of 1660. And that would be a very interesting and important contribution to the field of TB that we are planning to investigate. Okay, So I think that's all I wanted to say here. We predict that IS 6100 is moving and potentially could be used for sub lineage prediction, that is the evolution of certain strains, and potentially predict the genes that are regulated by this movement. Okay, so I would say TB, and a theme of this work is, I've told you about human, SARS-CoV-2, and TB. We partly picked these biological contexts because they are well studied by humans, and we had a way to benchmark and ground our predictions from NOMAD to orthogonal data and understand if our predictions made sense. And in all of the cases I've told you about, I think they have both made sense and extended or significantly extended what we know without using any ontology like a reference genome to make inference. And I think that's really exciting because it means that we can now trust the algorithm, or it gives us confidence that we may be able to trust the algorithm to apply it much more generally in contexts that we don't know so much about. We know that viruses and bacteria are increasingly detrimental to human health and evolving in ways that we don't understand, partly due to climate change and other environmental perturbations. And that is an extremely important direction that we're taking NOMAD in. Within human disease, I think there's a huge promise to be able to identify causes of non-Mendelian disease, including of tumors and other cases where we know that the reference genome is inaccurate and imprecise. Plant biology is another area that we're focusing on strongly because plants have enormous implications for crop, sustainability and food safety. And there are many other applications in DNA-seq and genome-wide association studies. And we're about to release the preprint of the human work in a day or two. So please check my lab's webpage or the bioRxiv for it. I'd like to thank everyone that was involved in this, including the land Stanford sits on, and take any questions.
Okay, Julia, thank you very much for a very interesting talk. Now the talk is open for questions. I mentioned you can raise your hand and I can give you the, you can ask directly to her. There is one question. Fernando Lopez Casillas, go ahead.
Hi, hello. Really interesting talk. Thank you very much. I'm not a mathematician. I barely understand all the foundations of your work, but I think what was clear to me is that NOMAD defines both the anchor and the target. Do you have any restriction on, or you impose any restriction to the algorithm to have a minimal size for the target?
That's a good question. So we use 27.
27.
Yeah, somewhat arbitrary.
Okay.
We could easily change it.
And then what are the minimum conditions or requirements that you have to let NOMAD define this sequence as a target? It has to be constant.
Oh, great question. Yes, we do have some thresholds. We have to see the target more than 5 times, I think.
Okay. And once you have that definition given, then you search all through the sequences available to
Exactly.
find potential targets.
Exactly.
Okay. And there you have a bias, or you have the program some requirements?
No, it is exhaustive search for every anchor and every target.
Okie dokie. Well, that seems to be kind of powerful and could be applied to basically any kind of sequence-based language. Have you tried that into accommodate that for linguistics? That's
a cool idea. I haven't thought about that. Yeah, I think that would be interesting. I haven't thought about that, but I agree with you. It could be for any language. I'm mostly focusing on biology, but you're right. It would be cool to do that.
Okay, well, thank you very much. Very,
Thank you.
illuminating talk. Thank you.
Another question? So I don't see questions in the chat. Well, in the meantime, more or less, I want to ask some questions. I have some questions. So the first one is in the analysis of the single cell transcriptomes, if you were able, using Nomad, to identify, like the case of the ribosome, the splicing RNA U6, that it has many copies of the genome, you were able to detect the transcripts of other repetitive elements, like transposable elements, that are quite similar. And if when you identified this transcript that comes from transposable elements, how did it influence the map that you have of the cells that comes from the tissue or from an organ? It has some differences, or nothing changed?
Well, that's very interesting. We have not dove into that. I can say yes, it does identify well, it identifies ALUs and some others. But have we dove into? We have not done the kind of manual scrutiny of what they mean and what they are. I think longer term, the goal of my group is going to be to develop some ways of automating this manual scrutiny that we currently have to do once we get a set of Nomad calls for interpretation, especially for cases where we don't know what the element is.
Okay, interesting. And the other question that I have is related to if you can apply this kind of algorithm to, for instance, chromatin modifications along the genome. So relating, for instance, some kind of language is also present at the level of the histone modifications along the genome.
Yes.
And all this data is available for many tissues, many different kinds of cells. I don't know if some kind of algorithm that is similar to Nomad can be used just to predict what happens with that genome of expression or
that is very interesting. Yeah, that's very interesting. And I guess that gets to the question Fernando was asking. I've described Nomad for the purpose of like analyzing raw sequencing reads, but you could equally well imagine taking, I imagine you're talking about, like, let's say something like single cell cut and run, or single cell ATAC-seq, something like that.
Yes.
Where you have you've run single cell ATAC-seq, let's say, or a special case
or DNA methylation, for instance.
Yes, let's say any of the, like, you basically run some algorithm that tells you, for this cell, here's the string of positions where I have a mark. You could take that data as a string and then run Nomad on it to infer patterns of sample, like patterns where the samples are not independent. So I think that is a really interesting idea, and could be done. Yeah, I hadn't thought about that.
Okay, that's great. I don't know, I don't see any other question. Well, just because I'm curious, but maybe I missed it. Maybe you mentioned that yesterday during the class or today, but from where comes Nomad, the name of Nomad? Why is that name?
It has some acronym, but I'm not sure what it is. Basically, I had some set of desires from names, but I then I allowed just like, but Caitlin and Tabor nominated the name Nomad, and I felt like that's a nice name, and they liked it.
Okay. So that's nice.
Yeah.
So,
can I ask another question?
Yeah, go ahead.
Well, first a comment. Nomad is the name of a very good meat restaurant in Monterrey, a city in Mexico.
Oh, it's a city
But
in
I don't know.
Mexico?
In Monterrey City is close to the border of the United States. But anyway, this is something that just came to my mind
Huh.
after hearing the name. The question is, in all these variants that you found for the ribosomal protein, once you have identified the target and the anchors, you really don't need to run NOMAD again. You could do, let's say, step-by-step manual sequencing to find whether or not your sample have either one of the targets or the other, right?
Yes, we don't run Nomad twice. It's only run once.
So NOMAD is just for discovery, and then once you have discovered the pair, you can analyze them by traditional ways.
Then we do a statistical microassembly, and then from that microassembly, which reduces by orders of magnitude, like at least 100-fold, probably a lot more in single cell data, then we align those sequences to a reference, for example, to understand them.
Okay. Did you ever try to find any physiological significance for the different targets in the ribosomal protein?
Physiological, did you say?
Yeah, like say, what happens if you have a variant regarding to the other one when you assemble a ribosome? Do they have different capacities to translate, to select?
Oh yeah, well, that's like a fantastic question. I would love to know the answer, but no, we have not done the hard work of doing experiments to experimentally determine the functions of the different variants.
Okay, and just one more, probably very out of the word question is, what happens if you try to use NOMAD to whole genomes of related species? Can you find phylogenetic
Of related who?
Can you find phylogenetic relationship if you try NOMAD on different species?
That's another super question. My guess is yes. And I think that's a really interesting direction to use it, to use it for
because I was thinking, there are some genes, for example, in the zebrafish genome that are not well annotated. And for some time they were really out of the radar, but because of the synteny, some of these genes could be identified. So probably applying NOMAD using a known target, a biased target, a biased anchor, you could find all those not so evident relations.
Yeah, I definitely think that's a very interesting idea, and I think it could be used. I didn't have a chance to tell you about how NOMAD definitely is able to discover paralogs within a genome. We did that with RNA-seq. For me, it's like a question of, I guess, yes, I think overall it's a very interesting idea and a way to basically, there's a way that we call this in the preprint, we call it compactors or targets by association. So if you have an anchor and a list of 10 targets and 8 of them are all aligning to gene number 1 and 2 of them don't have any annotation, then we kind of annotate those 2 targets likely as being from this gene that was, called by the first 2 targets.
Well, thank you very much, Julie.
Thank you. Any other questions? Well, NOMAD is it's freely available. I guess I didn't mention that. It's available for download. It's very lightweight. My student had a 2015 laptop where I think like less than 3 gigs of RAM and could run millions of sequences. So it's, please use it, ask questions on GitHub. And yeah.
Thank you. Is it very computer intensive?
No, that's the beauty of it. It's very, very lightweight. and we're about to release a version that is so lightweight that, it runs 100 gigs of data in 10 minutes with 30 gigs of RAM, which is just incredibly lightweight.
Good.
Was there one more question or no?
Yeah, there's one question from David Fuentes. Go ahead, David.
Yeah, thanks. I may have got a little lost during the. Can you hear me?
Yes.
Okay. So I might have got a little lost, but the question I wanted to ask is how do you define the what was the word?
Target?
False positives. How do you distinguish false positives from NOMAD, from false negatives, from other methods, from other method specific
Yeah, that's a
algorithms?
great question. We are in the process of doing that really rigorously, or trying to, with VDJ recombination. And so we're, it's a hard problem overall. I think all we have is basically inference, like to say, we tried it in this way, we benchmarked splicing, we looked at concordance between tissues. I think that's, or replicates, that's one way of convincing ourselves, I think, that we're not finding a false positive. Certainly with VDJ recombination, we can have kind of a definition of what's a true positive for NOMAD that is a false positive, a false negative for another algorithm. Because, are you familiar with VDJ recombination or antibodies?
Yeah, well, I had to. I'm part of the group you gave class yesterday. I had to study it, like for an essay we had to do.
Okay.
But yeah, it's like this mechanism that recombines the genome of somatic
Yes.
cells in the immune system.
Yes. So basically, long and short is there's a way to define, like, if NOMAD thinks this is VDJ, there's an orthogonal thing we can test and say, yes, it's probably really VDJ. And so that's another set of approaches to saying that this is a true positive for NOMAD and a false negative for other algorithms. It's a great question. I think it's a really fundamental question about doing science overall.
Right.
Yeah, I thought that maybe it would be like which ones have like a lower p-value or so, like the statistical methods. But
I don't trust. I mean, I think one of the ironies about being a statistician is I don't trust statistics.
Okay.
Well, thanks.
Thank you all. It was great to do this, and I really appreciate the invitation. I hope you have a wonderful evening.
Yeah, thank you very much.
Thank you.
Hopefully next time we'll be in person.
Yes, that would be wonderful.
Okay.
Take care.
Well, thank all of you for the assistance, and see you for the next one. Bye-bye.